Hello and good afternoon. Welcome to everyone joining us today for this SelectScience webinar titled Advanced Insights into Receptor Expression and Avidity Binding Dynamics on Cells Using Index 2 and heliXcyto in Drug Development. My name is Olivia Long and I will be moderating today's presentation, and I'm delighted to be joined by our guest speakers, Dr. Nichole Cerutti, Director of Biophysics at RoukenBio, and Dr. Vivien Hafner, responsible for scientific communication at Dynamic Biosensors. Please feel free to submit any questions you may have during the webinar today. To do this, simply head to the engagement window to the right of the auditorium and click to open the Q&A window. So without further delay, I am delighted to hand over to Dr. Nichole Cerutti and Dr. Vivien Hafner, and would like to thank them both for presenting to us today.
Today, Vivien and I will present the inducible cell line technology developed at RoukenBio and Brukers Biosensor's innovative single-cell interaction cytometry instrument. We'll show how the combination of the two enable deeper insights into how receptor expression can influence therapeutic affinity and avidity. We'll cover the technology, applications, as well as implications for advancing therapeutic development. Before we start, I'd like to introduce RoukenBio. Founded in 2015 as Antibody Analytics, we began by specializing in monoclonal antibody characterization, particularly for biosimilar developers. Our initial focus was on measuring antibody effector function in complex matrices, and as we grew, we integrated BioCore 8K systems, added advanced immunology services with primary human cell models, and launched our R&D department in 2019 to drive new bioanalytical capabilities. By 2020, we introduced cell line engineering and filed our first patents, which culminated in the launch of our Index 2 platform in 2023. Following our first external investment round, we very recently rebranded as RoukenBio, this September actually, to reflect our broader focus as a discovery-focused CRO. Our newly renovated Discovery Centre, which is located just outside Glasgow, houses just over 100 brilliant minds, offering a unique collaborative approach to drug discovery that's grounded in scientific empathy. Our services span immune cell assays, cell line engineering, safety and efficacy assessments, and mechanism of action studies, covering everything from immuno-oncology to inflammation and allergy. Measuring kinetics is essential for understanding how fast and how strongly molecules interact. These insights are crucial in therapeutic design. For simple interactions, the binding of two molecules is reversible and follows the law of mass action, governed by two key constants. The first, association rate, or K-on, which describes how quickly two molecules come together to form a complex. The second, the dissociation rate, or K-off, indicates how fast the molecules separate, and it's particularly important because it defines how long a therapeutic remains bound to its target. This duration, which is also known as residence time, can vary widely from seconds to even days, and has significant impact on therapeutic efficacy. Binding affinity reflects the strength of this interaction and is expressed as the dissociation constant, Kd. Kd is the concentration of a ligand that occupies half of the receptor sites at equilibrium, indicating the balance between binding and unbinding events. By convention, the receptor, A, is held at a fixed concentration while the ligand, B, varies. In drug development, understanding kinetics means going beyond just measuring how tightly a drug binds. You need to know how quickly it attaches, detaches, and then can repeat the cycle. Residence time is crucial as it influences the duration of a drug's effect and can even affect dosing schedules. Longer residence times may reduce dosing frequency, enhancing patient compliance and therapeutic benefit. Ultimately, kinetics allows us to design safer, more effective therapeutics by predicting how they behave in real biological conditions. Kinetics can be measured using a biosensor, which enables us to observe the speed and the stability of molecular interactions in real time. The video graphic above shows these kinetic events within a microfluidic channel, where you can see the real-time process of molecules binding and then gradually dissociating. One of the binding partners is immobilized on the sensor surface, and this we usually call the ligand, and the second partner is passed over the surface in solution. You can see how association and dissociation rates play out in a controlled environment, providing insights into how these interactions might behave in biological systems. In the graph below, we have a typical binding curve generated by the biosensor, showing the stages of an interaction. As the molecules encounter each other, we see the association phase, represented by the upward slope, followed by a steady state when equilibrium is reached. By replacing the analyte with buffer only, the dissociation phase then follows, and the bound molecules start to detach or unbind, shown by the downward slope. This phase allows us to measure the residence time, which is crucial for understanding how long a therapeutic will remain bound to its target, directly influencing its effectiveness. When we measure kinetics, we can see that the same affinity can actually result from different combinations of association and dissociation rates. For example, the molecule on the left binds very quickly and releases just as fast. However, the molecule on the right binds much more slowly and releases much more slowly. Both have the same binding affinity. However, they are governed by very different rates of binding. If we were to perform the titration experiment, we'd get an endpoint measurement that represents the overall binding strength or affinity. However, you can see in this approach, there's no way to distinguish differences in binding modes between the two molecules. Without measuring kinetics directly, we miss out on critical information that could influence a drug's efficacy, duration of action, and dosing strategy. So why are kinetics important for immunotherapies? The left graph shows the example of imatinib. The 4 circles represent data for binding the wild-type target protein and 3 imatinib-resistant mutants. All mutants have similar affinity as the wild-type for imatinib, whereas the off rate and the IC50s differ, showing that especially the off rate corresponds to efficacy of the drug. This has been further emphasized in a more recent study, showing that optimized binding kinetics can enhance drug selectivity and reduce off-target effects, leading to safer therapeutic profiles. The right graph shows the bell-shaped curve representing the duration of a T-cell receptor peptide MHC interaction and how this can influence T-cell response. You can clearly see that a very specific duration of interaction is critical for the most effective T-cell signaling and subsequent activation. Too long or too short half-lives can diminish T-cell responses. While traditional kinetic assays typically use isolated proteins or simplified systems, these don't always fully capture the complexity of a real cellular environment, where factors like receptor density, membrane and receptor dynamics, and cellular context can significantly impact binding behavior. By measuring kinetics on live cells, we can gain a more accurate picture of how a therapeutic interacts with its target under native conditions. This approach allows us to account for binding avidity, receptor clustering, and cell-specific factors that influence both the strength and duration of binding. Ultimately, cell-based kinetic measurements provide insights that are much closer to what we expect in actual biological systems, making this data highly relevant for predicting therapeutic efficacy, optimizing dosing, and minimizing off-target effects in real-world conditions. I'd like to now hand over to Vivien, who will talk you through Brukers Biosensor's groundbreaking technology, the heliXcyto, which is a first-in-class biosensor for the measurement of the kinetics of binding to cells. Following Vivien's discussion, I'll be sharing how RoukenBio have combined our Index 2 technology with single-cell interaction cytometry to investigate the effects of receptor density on the kinetics and avidity of binding.
Thank you very much, Nichole. So after this great introduction on why it is important to measure kinetics, I'm now very happy to introduce to you the tools you could use to perform these measurements. So as Nichole already explained, binding kinetics offer decisive information about your molecules, and this actually applies to many pivotal steps in the drug discovery process. And the tool I'm shortly going to present was developed by Dynamic Biosensors, and we now teamed up with the SPR team from Brukers and created the joint Brukers Biosensors business. And our combined portfolio now covers many steps from high throughput screening to the cellular assays I'm going to just now. So Nichole has already explained to you why it has a lot of advantages to measure kinetics directly on the cellular membrane and in the native environment. And I am now going to demonstrate the technology we use to do just that. And it's called single-cell interaction cytometry, or short scIC. And the biggest challenge we had to overcome was how to immobilize single cells in a microfluidic channel of a biosensor. So what you can see here is the biosensor chip, and the solution we came up for this challenge are these little polymer cages that are functionalized on the surface of our biosensor chips. We call them cell traps, and with these tiny cell traps, we can actually immobilize single cells on the chip surface in the microfluidic channel without the need of any capture molecules, and so these physically retain basically any type of cells. And on the left, you can see the heliXcyto biosensor, which is the instrument that actually performs all these measurements. In this short video, you can now see how these tiny cell traps are one by one filled up with single cells as they are flown across the sensor surface. And on the right, we have a second electrode that does not have any cell traps, and we can use that as a real-time reference. There are different versions of the cell trap chips available depending on your research needs, whether you want to look at the data from single cells only, or whether you want to perform a multi-cell measurement with 5 traps per chip. And then there's also different sizes of the traps available to accommodate the different types of cells. A typical scIC experimental workflow would look something like this. In the first picture, you see the microfluidic channel with the cell trap, and at first the cells are flushed in, as you have seen in the previous video, and the cells are trapped in the cell traps. You can control whether you trapped actually cells by looking at the snapshots that are taken by the instrument automatically, so you can see whether all the traps were filled with single cells. Next, we flow in the fluorescently labeled analyte molecules, and upon binding of these fluorescently labeled molecules, the fluorescent signal will increase, and this is measured in real time. Afterwards, we inject buffer, and this will initiate the dissociation of the analytes from the cell, and this can be seen as a decrease in fluorescent signal. And after that, in the last step, the flow can be reversed, so the cell is pushed out again of the cell trap, and the chip can be used for another measurement run. So from these fluorescent measurements, you can then derive the association rate as well as the dissociation rate. I would like to briefly compare the scIC technology to other technologies. First of all, surface plasmon resonance, SPR, is very established for molecule-molecule interactions. However, it is challenging to adapt this technology to cell-molecule interaction measurements because it is difficult to stably immobilize the cells on the sensor surface, and also the detection depth is limited. So you will always see the signal coming from the lower part of the cell only, instead of from the full cell as we have it for scIC. Flow cytometry, on the other hand, is very feasible for cell measurements, but not as feasible for molecular interaction measurements. Flow cytometry is a single time point measurement, so you cannot measure real-time association and dissociation rates with it. So now let me show you some application examples of kinetics on cells. I would like to start with a very prominent example from the field of immunotherapies, that is immune checkpoint inhibitors, or in this case, it's nivolumab. So immune checkpoint inhibitors work by blocking the interaction on T cells with immune checkpoint receptors and thus retaining their activity and retaining anti-tumor immunity. And you can see here on the left side a typical sensorgram with two different concentrations of fluorescently labeled nivolumab that was binding directly to the cell surface of the T cells. And then you can see the dissociation in blue. And what you can already appreciate by eye is that this dissociation curve shows two distinct phases, a faster one at the beginning and a more slower one at the end. And if we fit this data, we can also confirm that there are two phases, as we see that a monophasic fit clearly deviates from the data, whereas the biphasic fit matches much better to the dissociation curve. So what this actually shows us is that there are two modes of binding happening during the dissociation. We have the faster dissociation due to the affinity of the antibody, but then we also have a more stable interaction with a slower dissociation that can be derived from the avidity of an antibody binding with two arms at the same time. And this has very important implications for the clinical efficacy of immune checkpoint inhibitors, as they need to have a specific dwell time on the cell surface in order to actually execute their therapeutic effects. So let's look at a more complex example, that is target receptors that are difficult to express. Many of the most interesting targets are actually multi-transmembrane domain proteins, for example GPCRs or also CD20. And these proteins are very hard to isolate and then refold in order to analyze them outside of their membrane context. So this also shows the beauty of the scIC technology, because you don't need to isolate these transmembrane proteins anymore, but you can investigate their interactions in their native environment within the membrane context without any preparatory steps. So in the first example, we looked at a small molecule binding to a GPCR. You see in a first assay development setup how we looked at the specificity of the interaction. The blue curve shows the interaction on a cell line that does not express the target GPCR, and accordingly we don't see any signal. Whereas the orange graph shows the interaction on the target expressing cell line. Now in the next step, you can further characterize the interaction by doing a multi-concentration titration and looking at the full kinetics to derive association-dissociation rates. In the second example, we looked at a very prominent therapeutic target, that is CD20. Here we labeled rituximab, a therapeutic antibody, and we looked at the binding of rituximab to CD20 directly on the surface of the B-cell line. Now, for even more complex situations, you can also make use of the dual color assays in the heliXcyto. You can measure two colors at the same time, so you can also mix different antibodies labeled with a green and a red fluorescent dye and look at them simultaneously, look how they cooperate, how they can influence each other's binding to the surface targets. So in this example, we used an anti-HLA antibody as well as rituximab, and you can see the different kinetics simultaneously, one in green and one in red. And you can clearly appreciate here how the binding kinetics differentiate from each other. So with this, I hope I have given you a good overview of how scIC works and what type of applications are possible. And I am now happy to hand over back to Nichole, who's going to show you how they used scIC in combination with their unique platform for titratable expression.
One of the key challenges in developing tumor-targeting therapeutics lies in identifying optimal tumor-associated antigens. Often, these target antigens aren't cancer-specific. They are overexpressed on cancerous tissue, but can also be found in healthy tissues. This heterogeneous tumor antigen expression not only makes efficient treatment more difficult, but also contributes to immune evasion by tumors that downregulate the expression of the target antigen, so-called antigen escape. This challenge applies to multiple immuno-oncology therapeutic modalities, including immune cell therapies, immune cell engagers, ADCs, and conventional antibodies. The expression of target antigens in normal tissues can lead to off-tumor toxicity, which is a common side effect associated with many immune-engaging therapeutics. In particular, on-target off-tumor toxicity has become a significant concern, and regulatory guidelines have been established to assess the undesired targeting of healthy tissues that express the target antigen, as you can see here in a snapshot from the current FDA guidelines for the development of CAR T-cell therapeutics. The conventional approach to assess the impact of antigen density on the efficacy of therapeutics is the use of a panel of cancer cell lines that express differential levels of the antigen of interest. However, there are multiple limitations to this approach, including cost, access, expression range, and cell background. RoukenBio has developed a solution to this, the Index 2 system, a platform which utilizes chemical proximity systems for the inducible and titratable expression of one or two target antigens in a controlled manner. We've built our Index 2 system using TOK1s as the background, introduced an induction cassette, and a second construct containing the target of all targets of interest via lentivirus. To exemplify the system, we've used a number of target antigen examples and have demonstrated, as you can see in the graphs, that we're able to control the expression of these antigens over a wide expression range by varying the concentration of inducer. You can also see in the graph on the right that we're also able to simultaneously induce expression of two different antigens, also at varying levels, by controlling the concentration of inducer, CD22 under the control of indole acetic acid system and CD19 under the control of the abscisic acid system. Using the single HER2 expressing cell line, we sought to investigate the effects of target density on the kinetics of trastuzumab binding. We chose 4 differently expressing HER2 cell lines and quantified the expression of receptors using flow cytometry. We used cells expressing receptor at low, medium, and high levels, and we fixed these cells before analysis to ensure we prevented any receptor internalization during binding. Fluorescently labeled trastuzumab was passed over captured cells at 3 different concentrations, starting at 1 nanomolar and ending at 25 nanomolar. And we followed this by an extended injection of buffer to follow dissociation. Trastuzumab was able to bind to all 4 HER2 expressing cell lines, and controls included a wild type control and a non-induced control. And you can see in both these controls that there's little to no binding of trastuzumab. What you can also clearly begin to see by eye when just looking at the traces is that there seems to be a difference in the off rates, with trastuzumab being bound for longer periods of time to the cells with the highest number of receptors as compared to those cells that have a lower number of receptors. When normalizing the fluorescent signal to the number of cells, we can see that the amplitude of the signal observed directly relates to the number of receptors per cell. More receptors means more binding sites for trastuzumab, which in turn means a higher signal is measured. When we fit the data to a 1-to-1 Langmuir model, we find the KDs measured are all in the picomolar range. Cells with the lowest receptor number have the highest KD and therefore the lowest affinity, whereas cells that have the highest receptor numbers have the lowest KD and therefore the highest affinity. This is where the kinetics becomes quite important. The on rates measured are all very similar for all 4 cell types. The off rates are driving the differences. The lower the receptor number, the faster trastuzumab dissociates from the surface. Residence time is 10 times higher for cells with the highest number of receptors, which strongly suggests avidity is playing a role in the binding at the higher receptor numbers, as compared to those cells with lower receptor numbers. As we look ahead, this technology presents exciting opportunities for expanding our research applications. One area is receptor internalization and recycling studies, where we can observe how long a therapeutic remains effective on the cell surface before the receptor is internalized or recycled. This can be done using pH-sensitive dyes, which change their fluorescence upon internalization and exposure to different pH levels. Understanding these dynamics is key for optimizing therapeutic duration, especially for targets with rapid cycling or high internalization rates. Another promising outlook is the use of our dual expressing cell line with titratable expression to be able to study bispecific binders. By adjusting the receptor expression, we can evaluate how bispecific binders engage with both targets under various expression levels. And this will ultimately provide valuable insights into binding efficiency, selectivity, and therapeutic potential. These applications will open newer possibilities for designing more sophisticated, durable therapeutics with targeted engagement strategies. In summary, we've shown that scIC measures direct binding to targets in their native environment on cells, and we're able to measure both kinetics and avidity. We've also shown that target density influences the kinetics and avidity of therapeutic binding, and there is expanded applicability in the areas of internalization, recycling, and even competition experiments. Thank you very much for your attention.
Thank you very much to you both, Dr. Nichole Cerutti and Dr. Vivien Hafner, for your wonderful presentation. I hope that everyone listening enjoyed the webinar and found it as informative as I have. We will now move on to the last part of today's webinar, the question and answer session, the Q&A. As a reminder, to ask a question, you just need to simply head to the engagement window to the right of the auditorium and click to open the Q&A window. So to kick off this final portion of today's event, my first question is for Vivien. Which cell types can be used in interaction cytometry?
Thank you, Olivia. Good question. Actually, you can use any type of cell in scIC. You can use adherent cells as well as suspension culture cells, and you can also use fixed cells or living cells. It really depends. So any eukaryotic cell type. We cannot measure bacterial cells at the moment. You could use them as an analyte, but not trap them in the cell traps. But any eukaryotic cell type would work out well.
That's great, thank you. I also have another question for you, Vivien. What is the throughput of the instrument?
That also highly depends actually on the interaction you want to look at, of course. So depending on the association, dissociation, how long you need to measure to really capture that. But as a general rule of thumb, I would say that we would need like about 1.5 hours for a one. Of course, this is not the hands-on time. Hands-on time will be about 30 minutes. The rest will be automated. So you can set up your assays and let the instrument run overnight and run overnight several measurements, which will add up to approximately 10 to 15 measurements for 24 hours. So this is not a high throughput instrument for screenings, but it's more for the in-depth characterization on cells. And yeah, that would be about the throughput, but it really depends on how fast your kinetics are. If you have really weak binders, you can probably even ramp up to higher numbers for overnight measurements. Yeah.
Nice, lovely, thank you. And I now have a question for you, Nichole. Is the data generated comparable to other binding methods?
Yes, absolutely. We've tested the SeIC, looked at SeIC with binding to therapeutics using recombinant proteins on SPR, surface plasmon resonance biosensor chips. And we were able to get very comparable data when using the two different techniques. As Vivien has mentioned, it's really a throughput issue, as we do tend to see a bit of a longer residence time with the cells, and that's obviously due to the native environment that the targets are found in when doing the ScIC as opposed to the SPR experiments.
Lovely, thank you. And now for Vivien again, what is the detection limit for interaction cytometry?
Regarding like the receptors you need per cell, we haven't really hit the lowest limit yet. So if you have any challenging examples, please contact us and we are happy to test it out. But also a rule of thumb would be that you would need to have about 1,000 receptors and targets per cell. That's definitely, we have a comparison to flow cytometry, for example. If you measure KDs with flow cytometry, we are much more sensitive. We can measure at much lower concentrations. So yeah, about 1,000 receptors per cell. But as I said, we haven't really had an example where the expression was so low that we can actually put our thumb on a specific number in the lower limit.
Nice, thank you. And then for you, Nichole, this question says, is this type of methodology accepted by regulatory agencies?
That's a good question. And the overall answer is it's not really been submitted as yet. However, there's data that's been collected using the instruments to underscore important qualities such as robustness, reliability, and reproducibility when it comes to qualifying assays. And I think more and more, because we've been able to demonstrate the importance of target density and the requirements of the FDA to have this kind of data, it's very likely that this kind of technology will soon be required for IND submissions.
Lovely, thank you. I have another one for Vivien. What is the detection limit for scIC?
I think for the analytes, of course, depends on the KD. For the receptors per cell, as I said, for the lower limit, we don't really have a specific number yet. We usually work with about 1,000 receptors per cell as a lower limit, and that's what you can definitely try out. But we are happy to also try out lower expressing cell lines and see what results this can give.
Lovely, thank you. And another question here. Why is it so important to understand target density?
So, as I had mentioned earlier in the slides, healthy versus cancerous tissue express targets at very different levels. They can either be overexpressed or underexpressed, and in order to prevent on-target off-tumor effects, we need to understand the role that the density of our target plays in the therapeutic efficacy, and so that you can then understand why it is so important to be able to replicate this in an in vitro setting so that we can understand how the therapeutic is going to work in an in vivo environment.